we'll just go ahead and get going and let Dr. Murdoch tell us. She's got some questions that we sent in, and then she's also got the background knowledge of what she does that she can share what we don't understand or don't know. Or so Dr. Murdoch, we'll just let you take over.
Okay. Well, I wasn't sure if this was going to be like a Q&A or what exactly. I didn't really prepare a whole lot. I suppose I could.
I have questions if you'd like to start that way. And, you know, perhaps the questions will lead you into explaining what we don't know, because I read your article, Eddie, you sent it out, the research on the terminal sires breeds in the US. And, right. You know, to the best of my ability to understand it, I got a few questions from that. So if you'd like, we can start that way there.
Sure.
So rather than, you know, asking you for, you know, to wing it on a presentation there. My first question was, I noticed in that article, the numbers of sheep for which you did, I think the 50K chip ranged from 11 to 46. And then there were others from, I assume, different studies, other genomes. I know my ability to pick the right word is, you know, I'm just really kind of guessing here. What's the optimum number? How many would we want to do if we could do as many as would be optimum for this?
Well, that I'm probably not the best to answer that, and because in my perspective, it's good to have them all, right? The more, it's almost always the more the merrier. Right, but okay. It really just depends on what the question is. So part of what we did in that study was to say, okay, how many different breeds can we collect? And we actually collected a few more, but we said, okay, if there's less than 10 or and unrelated animals, then we can't in all good conscience put them in this study. So we started off with a few or more, with a few less, and then we tried to gather some more information and gather more animals in that particular case so that we could look at them. We actually did genotype more, but because there were not enough individuals to be to be included, so we left them out, right? And you have to think about in all of those scenarios, and in that case, right, how much of the breed is that really representative of? You know, so we always have to be careful there, you know, and getting a little bit to what you guys, or what I was thinking you guys wanted in just some of these questions that I kind of asked, is you do want to make sure that you're genetically covering as much territory as you can. So, you know, you want to have, if you had, say, for example, I'll just give you a scenario of a family or a flock that were highly related, you might be able to get information about that, but that would not be representative of the entire breed, right? It would be representative of that flock. So depending on what exactly you guys wanted to do. And so in that study, I used the 50K thing. And so I also developed the Flock 54. And okay. The idea that I had in mind there when I did the Flock 54 is, you know, and part of this goes a little bit just because I know the technology. So I was part of the team that developed the first 50K SNP chip. So I know what goes on in that information and the intent of that. And so being a scientist that does that, it kind of saddens me a little bit to see a bunch of companies charging producers, you know, $15 for this and $18 for that and $15 for this. And I'm like, you know, it was fine when we first started doing this kind of technology in other breeds, mostly in other breeds, because it first started kind of in dairy, in cattle. You know, when there was 1 or 2 tests, okay, you can charge people $15 for a cow or a bull, right? But in today's technology, there's no reason why, if we don't just if we change platforms, and that's what I did with the Flock 54, we change platforms in how we do the test, we can do them all for very cheap. And so that Flock 54 really looks at 1,000 markers for $18, right? And part of that was my own experience, right? So we have some sheep at University of Idaho. I'm like, wait, you're going to charge me $18 for parentage, and then another $15 for scrapie, and then another $13 for spider lamb. I'm like, this is crazy. And I understand why they're doing it. They're doing it because when they run it as a single test, that's what it costs them, right?
Awesome. Right.
But if we change technology and put it all together, then we can do that and get a lot more data for much cheaper. So that's essentially what I did with the Flock 54. So the 50K test is more money. It's $25, $30, $35.
Right.
And that actually in the last couple of years has come down, because it was probably about 2 years ago, they were charging $90 a sample for that, right? So who can afford to pay $90 a sample? So that's why we did the Flock 54, right? Why pay $90 when a lot of that information, it can be helpful, but it's not really what most producers need. So what is the minimum of what most producers need? And so that was the idea with the Flock 54. So my thought was, and again, this was just a thought, depending on what you guys wanted to do, was to take a representative group of animals. And when I say representative, I mean, you want it to be representative of the genetics of the breed. So you want to try and capture that diversity. So test these animals on the 50K chip, and then also test them on that cheaper Flock 54 and say, okay, this is the information we get on the 50K, and then this is the information that we get on the lower density genetic information. Is that lower density going to be sufficient and give us the same kind of detail, right? The more there's another chip out there that's 600 markers that costs $120, right? So you can keep going up, and the price tag keeps going up. So my thought is this Flock 54 gonna have enough resolution to help you as a breed association to do what you want to do with this, right? And so that was I don't know what the answer is. The answer is, it's gonna be yes or it's gonna be no. Or maybe even somewhere in between, but that was my thought, is will this Flock, if we ran the same animals on both of those tests, would we be able to determine breed? I know we can determine inbreeding coefficient. Would it be enough on that lower density panel as it is on the higher density panel that I used in the paper? So that was my thought.
For testing the same sheep using both tests, how many of those would we want to do that way? A dozen, 6, 50, up to 30? Well, what are we talking about there?
I think in the original email, I tried to budget around 50.
Okay.
Again,
Okay.
It goes back to that magic number. More is always better, right? If we did them all, then we would know them all, right? But it really just depends on what you guys want out of these types of products, and how can I be helpful and beneficial that way.
So we'd be doing 50 sheep using both the 50K and the Flock 54?
Right.
Okay. And
that
then
was
as a possibility. And then is it future sheep that we might be able to do using the Flock 54?
Right.
Okay.
So if it turns out that we can get very similar data from the Flock 54 than we can with the 50K chip, and if we can I just did a quick calculation, I don't know if it's, so, it would cost us, basically $16,000 to do all 1,000, to do the entire flock. We would have genetics on the entire flock for $16,000 with the flock 54. And with some research project money, that could actually be not that hard to do. If it was $30 for a 50K chip, so $30, you know, times 1,000, then you're looking at about twice as much money, right? $30,000 to do. And that would be to do all 1,000. That would be the breadth of what you're saying your U base is, and you would have that genetics on it. And so again, really ultimately is what do you want to, you know, to me a little bit is what do you want to do with the data, right? So when I get back to that, if the only thing I ever care about is scrapey data, then maybe that $13 test is my best bet. But if I want scrapey data, spider lamb, callipege, I want some other things in there, some hold horn markers in there, then maybe I want to pay that $16 and have that much bigger, right? Depending on what you want to do, maybe you want to pay the $30 and have the full 50K run on it, right? It's really what you guys want to do.
Okay. Well, if we end up doing all the registered sheep, that makes life easier, but in a way. But if we don't, how do you best select diversity? I mean, do you randomly go through the registered animals and randomly select? Do you look for geographically separated flocks? You know, do you look for phenotypic diversity? You know, your big sheep, your little sheep, your white ones, your colored ones. How do you get the widest spread of the genetics?
Yeah, that's a great question. You want to do a little of all of that, right? So you do want to often geographical distance is also related to genetic distance because they're breeding within the folks, you know, that's their breed base and that's as far as they go. So you do want to cover that breadth. You really want to, in order to cover your genetic diversity, you want to sample those more distant and have, or at least have representation of those more distantly related animals, whether that, that's not exclusive of geographics, right? So I think the one thing that we could see is the sheep, the Suffolk sheep that we have at the University of Idaho have been in the Northwest for a long time. They do not have as much hemp in them like the Suffolk sheep that are in the Midwest, right? And so they look different and they are genetically different. We can still call them all Suffolk if you want to call them all Suffolk, right? They're still going to be black-faced sheep. There's still some similarity. They are still a Suffolk. They're not a Rambouillet. They're not another, but they are different because they've been isolated and been in different and bred in different. Those genetics have been isolated for so long. Some of those phenotypic differences are actually a result of genetic differences. So sampling those phenotypic differences is actually, if you want to cover your entire breed base, you do want to have some of those different animals in there. You know, one strategy that the horse community does is they actually just do parentage on all of their animals, and then once they have that, you just, when you go ahead and re-register, you just say what the breeding pair was and prove, and then you sample that new individual, and then they have the genetic basis from all of the other in their parentage profile, and then it's and you can do that quite inexpensively. The Flock 54 will do that easily. So it really just depends on what you want to do, right? If you want to start moving into having a little bit more resolution for finding associated traits, then you do want to move a little bit, think about more of that 50K product. It really just depends on what you want to do as a breed. Right, so that's, I mean, the 50K product works well. So really just depending on what it is you want to, as a breed association, what you want to do.
I'm not sure I followed what you were saying. So we would want to use the 50K if we were also looking for how different traits associated with each other. In other words, I don't know what are good traits, but let's say scrapie and weaning weights. So we'd want that to, to see the linkage between those two, or we would need the 50K. Is
Right.
that
So,
right?
so the higher density chips will do better in that. And it's not necessarily
Okay.
even 2 traits, but if you want to, say you want to make genetic improvements on weaning weight, right? What we do then and how we do that is we run 50K chip or higher to find the genetic relationship with that trait so that we can say, oh, okay, this is what you're looking for and this is what you want to select for.
Okay. Okay. All right. That makes sense.
So in some traits can be correlated. I wouldn't think scrapie and weight necessarily is, but certainly birth weight and
Right.
weaning weight and those kind of things are correlated things. But if you're wanting to make a bunch of genetic progress on those types of traits, especially if you don't already have that information, then the 50K product becomes much more valuable.
Okay.
It really just kind of
Okay.
depends on what you guys want to do with, I mean, you can go all the way up to sequence level, depending on what you want to do and what questions you want to use this technology to answer.
Okay. I think our primary goal, and Eddie, you can tell me if I'm wrong on this, the primary goal for us was to be able to test a sheep that we think is a purebred St. Croix, but is not registered, and say, yes, genetically it falls within that circle of what we defined as registered sheep, and bring sheep back into the registered fold just to increase the gene pool. I think, am I right, Eddie? Is that a fair representation of what at least our initial goal was?
Yeah, I started to ask that question. I got sidetracked a while ago. We would have Barbados and other hair sheep to see if we're cleaning them or if we've got any pollution from them. I know you already have data on Dorpers and Katahdins, but is that something we'll have to do as well to see how much correlation there is between other breeds and our sheep?
That is, I think that could be beneficial, but I think you want to be a little bit careful there. Because sometimes you're going to find some things that are somewhat correlated, I don't know. So if I'm looking for areas that can, they're all hair sheep. So the areas that control the genome that make hair sheep are all going to be the same in that particular area, right?
Okay.
So I think, and there's going to be some similarities, but depending on how well you group and define things, you can actually, start to pick out some differences a little bit.
So Dorpers and Katahdins have sort of a breed profile already. Is that correct? Because that was one of my other questions is, if we have the St. Croix done in isolation and we have that nice little circle or oval, do we know anything if we don't know any of the other breeds that could be confused with the St. Croix? In other words, if we don't know what a Katahdin looks like, how do we know that it's, well, I guess it would still fall out of the circle then.
It will still fall. It will still fall out of the circle, and so I mean that circle can take different shapes. So remember, a Katahdin has way more genetic diversity than your St. Croix, right?
Okay.
It's going to have can maybe yeah, maybe I won't show that. But St. Croix is so we can ask and do things a little bit differently. What you saw there in that paper was to look at genetic similarities based on some genetic information, but there's other criteria rather than just the way I displayed it there in that PCA plot that we can look at. And that is the amount of heterozygosity, right? So St. Croix are much less heterozygous because they've kept true in their breed, whereas, you know, Katahdin, you're going to have some overlap, but they have much more, I guess what we call heterozygosity, more diversity in their genome than what the St. Croix does. So there's a couple different tests that we can use to ask those questions.
Okay. Just out of curiosity, where do Dorpers fit in that heterozygosity, homozygosity kind of, you know, line? Are they a mix also? Do you happen to know?
I don't know off the top of my head. I'm just looking at some data here, but it doesn't particularly have that one.
That's okay. It's pretty idle curiosity, I guess.
Yeah, you know, and you're right in the sense that, you know, comparing to those outgroups helps you to form your sort of or understand the I'll say the connectivity or the relationship within your cluster. In understanding some of that, it can be important. But I think in the end, that is not really the defining factor, particularly for St. Croix. And the reason why I say that is because they are already a tight bunch. They're already a very tight cluster, right? So we'll run into more trouble in a breed that's not a tight cluster. So like a composite breed or composite breed that's been made up of a bunch of different breeds, then you have a much harder time defining those because you will have way more overlap. And so some of the information that I've seen coming through actually from from some of the St. Croix genetics that I've looked at, you're a fairly small cluster, very tight cluster already. So it'll be, you know, it'll be how. So if you have, I guess, if you have a cluster of dots, and you think about that data, right, how far are you willing to accept that genetics coming in? That's a you guys' question, right? I mean, we'll just run the genetics and then it's up to you guys and to say, because, you know, it could be that other animals, you know, still have a little bit of something left in them or a little bit, you know, that snuck in the genetics there somehow. Right. And so defining that and that tolerance, that's all up to you guys. And that's where you can also then rely on that parentage information. So you can say, okay, well, I have a small little out cluster here, but we agree that that is still a part of what we want to do. And then you just have to show relationship to that, you know, little side cluster as a part of the big cluster, right? Those are all
Okay.
you guys' choices, right? And it, yeah, and once you define your cluster within your your breed is not so big that you can't literally sample the entire association or your entire breed base if you wanted to, right? And then it just becomes you know, okay, somebody else wants to ask this question, how related are they? Then we can sample that thing and say, yeah, you're right smack in the middle, or no, you're a small out cluster, but still pretty close or whatever. Then you guys make those decisions, you know, not me. I just run the genetics. The data are what the data are, right? And then you guys define how you want how big of an acceptance you want to do in there.
Okay. Okay. I think that answered the next question I had about, you know, the confidence level that you have if you test a sample and then you test an unknown sheep. The confidence level depends on how big you make that circle, I guess. In other words, you test the unknown sheep and say, oh, yes, this is definitely a St. Croix, or maybe it is, probably it is, but we're not 100% sure. we're only 85% sure. But I guess it depends on whether you make your circle very small or if you make it a little bit bigger.
Right.
So, okay.
I mean, and you can think about, right, when you think about the data, I didn't even pull that paper up. But when you think about those shapes of those clusters, they do, stretch out, and they can be, and then, that is the genetic basis or the genetic sound, right? And people
Right.
can determine that a little bit, how they want. So I guess the point there is, depending on that breed base, they can be very tight or they can be more spread out, depending on how much heterozygosity and how much other mixing of things are in there a little bit.
Okay. Okay. So I guess another question I had was that if you have a sheep and one parent isn't a St. Croix, it's something entirely different, it's a wool sheep or whatever, and you take those offspring and you continually breed them back to purebred St. Croix, how many generations would you go through before you can no longer distinguish the offspring as not St. Croix? In other words, I'm assuming that if you took a cross, that the first offspring would probably not look genetically like a purebred St. Croix. But if you keep breeding it back to a purebred St. Croix, how long until you can't tell anymore that there was something mixed in there, 3 generations ago or 5 generations ago?
That's a good question, and goes back to how hard you're looking and how much similarity. But in general, 4 generations and you have to look pretty hard. 5 generations and it's very, very hard to find any differences after 5 generations. So even 4 generations, you have to look hard to find. I think if I remember correctly, it's going to be about 5% at 4 generations. If you're back crossing to that breed within 4 generations, you will pretty much be there. And you have to look hard to find that you're not. In 5 generations, you will almost entirely be there.
Okay. Okay. Eddie, what brought that up was the upbreeding program the Katahdins do. That made me wonder how quickly that could happen. So, okay.
Yeah, a number of cattle breeds do that and get them up to 15, 16. They're pure.
Okay.
They're pure, not full blood. Pure. Those NSIP
right,
accepts flock 54 data.
No. And you're saying NSIP?
Yes.
No, I do not believe so. I mean so when you say for a genomically enhanced breeding program, is that what you're asking?
We met with Rusty Burkett, and he was saying that we could do that and greatly enhance the accuracy of EBVs with 50K, and that's why we're trying to wonder, could those two match together in both the study of the breed and in improving. We don't have anybody in NSIP, but we may in time.
Yeah, so NSIP up until, well, even now, right now, they do not deliver genomically enhanced breeding values, but they will very, very soon. And those genomically enhanced breeding values are based off of a 50K chip. So the flock 54 will not be what they want for that particular thing. They want a 50K chip or higher.
Okay.
I'm sure they wouldn't say no to higher, but that would be the minimum that they would want.
And our understanding was that the NSIP, the owner of the sheep owns the data. So any sheep that had a 50K chip done for NSIP, we could use that genetic information also as a breed representative for the numbers for our testing purposes. Do you see a problem with that, or is that your understanding too? It's the same test, basically.
Yeah.
Okay.
You're going to pay for the data, so you should own it,
Right,
right? NSIP does not pay for the data.
right. That was exactly what they said. It belongs to the owner of the sheep, so that
That's
makes
right.
sense.
So you're gonna pay for that. Right now it's about $30, maybe $25. You can get it maybe down to $25. So you pay for it, you collect the phenotypes, and then you can put it in and become a member of NSIP and put it in there and then start to collect breeding values for sure. But yeah, you own the data is yours. You own the data, or you could, if you wanted to, as a breed association, if you had other funds or something like that, where then the breed owns it, right? When I think about what does the Angus Society do, right? They, as a society, they actually subsidize some of this stuff for their memberships.
Right.
And then as a society, they help process this data for their membership. And then the individual itself, because the breed association pays for that, it is the individual's data, but they don't really just get the data back per se. And a lot of that is because what are they going to do with it, right? They don't know how to process it. And in some ways, especially when you talk about a breed association, it's more valuable in context of with other sets of data, as opposed to I have an N of 10, and this is what I have. But the information within that data, even in those breed associations, that information then gets used by the association, and then also to the producer themselves. And I think the association in the cattle industry, they're pretty tight on how and what they use that data for. And they do usually hold a pretty good confidentiality for that, right? Because remember, these data can be useful to detect recessive diseases, right? And so the association themselves, they want to know so that they can help producers get rid of a problem if there's a problem, but not all producers want all other producers to know about that problem, right? So that association does hold that within confidence within their association, and then reach out to those individual producers and work with them. So it really, again, it really just depends on what you guys as an association really want to do. The genomically enhanced breeding values and the NSIP, they're not quite at the stage where they will maybe give you the disease, recessive disease things that you probably get. I think we're going to give maybe 3 traits back on that chip. And those 3 traits may not even be relevant that much to St. Croix anyways. But
okay.
So hopefully that gives you a little bit. So NSIP is really meant about enhancing and using the genomic tools to enhance their breeding values. Yeah, and so it becomes very powerful if you enter and keep all of that phenotypic data. You can do it that way. But
okay. Well, NSIP has its goals and its purposes. For what we were looking at, where we're looking at using the genetic profile to identify sheep that have somehow lost their registration or their ancestry, really. Have you seen any other associations use genetic information that way? And have you seen bad things happen, what kind of pitfalls there are? Or conversely, have you seen benefits to associations once they have a genetic profile? How has this data been used by others in your experience?
Well, I think I just gave you a pretty good example of what the Angus Society does. They also use it for breeding values. In that case, the society themselves run those things. But remember, those are bigger societies, right?
Right.
They have more resources and more money. And so your question, is there pitfalls?
Well, given that our goal initially, at least, was to bring more sheep into the gene pool of that, St. Croix sheep back into the gene pool, does that happen? Or, are there reasons why it won't work? In other words, we won't be able to successfully identify St. Croix sheep that have lost their registration, basically, or anything where it turns out not to do what we hope it will do.
So I don't know. I mean, I don't think really there's any sheep producers that do this kind of things. I do think there's a number of other livestock associations that do things like that. I mean, ultimately, and this is what I tell my genetics class is, it will tell you the truth, but do you really want to know the truth? Those are the pitfalls, right? You really
Right.
kind of have to be a little bit careful on how you handle that data, how you deliver different data, and that is really more of a personnel sort of association problem. And then there's also, so the one scenario that I've seen happen a couple times in sheep, but more so in horses, is that you can have sometimes a different sort of biological consequence in breeding or pairing, and we call it a chimerism, where an animal doesn't appear to be what it actually is, like the genetics doesn't match, and that's because that animal actually has a cross within itself. It's a chimer of two different units, and so that kind of information can be a little bit confusing. And I know that in horse registries, it's like I have a video of this mare giving birth. This has to be, the parentage has to match. And it turns out that there were twins in there, and then one got reabsorbed in the other one, and there was blood crossing, therefore DNA crossing, therefore crossing of information. So that those kind of little glitches can kind of happen.
Okay. Okay.
And so overall in genetic testing, I think, well, even our own flock, it's just like, oh, okay, we're sorting rams yesterday, and I'm like, okay, that one's got to go. It's a spider lamb carrier. And they're like, wait, we've tested these for however many years. I'm like, yeah, you tested the sires, and this ewe has never dropped a carrier before, so we didn't think, but nobody actually tested her. But now that she has dropped a carrier, it doesn't mean you have to get rid of that ewe, but she's a carrier, so you better watch out every time you breed her and just make sure. So again, they're tools for people to make decisions. The decisions are still up to the people and how they want to use that data and manage that data, right? So we'll probably end up calling that ewe, just because she's been around, and we had called when we first tested. We did all the rams and all the lambs, and then just kept kind of all the newer generations. So this was an older ewe, and we did not see her. None of her previous offspring had been identified as a carrier before, and we know the ram is not. So we know she is.
It had to be.
Yeah.
Okay.
So sometimes, people don't want to test because they, because
you almost don't want to know.
Be a little bit complicated, but in the end, it is more information for you to make the decisions you want to make, and we make those decisions whether we do it blindly or whether we do it informed. So yeah, and you know, my thought was
Sure.
just to test that 50K versus the flock 54 and to see if the lower density would give you enough information so that you could go broader with your breed base. That was my original thought, but it really just depends on, there's all sorts of different tools. So it really just depends on what all you want to get out of this data. Yeah, so 50K is a decent option, so. It's just
Okay. Practically speaking, Jason, I think is saying he has to leave though, but he'll be listening to the recording. I guess practically speaking, what you would be helping us do is decide which, how to sample our animals, which animals we would at least start with to determine if the 50K and the flock 54 would give us, enough information with the flock 54, and then whether we widened our testing if we, depending on what we can afford, I guess, basically. And then you would interpret the data and help us get that circle. I looked at the graph and it says it's comparing eigenvalues. So I looked up eigenvalues and I still have absolutely no idea what they are, but I'm trying to figure that out. But that's what you could do is interpret that data and kind of give us that cluster and tell us, this is what fits in, this is where all the dots fell. Is that right? Is that your role?
Right.
As
Yeah, let me see. I have a lot of things open here, but you can see my screen now, right?
Yes.
Okay, so I just asked my grad student to just send me some information really quickly for you guys to just take a look at. And so she just dropped some data slides here.
Okay. Wow.
And so I just want to share this with you a little bit, and then we can get back to my role. Okay, so this is when this is whole genome sequencing. So when I look at that's more than the 50K. When I look at the number of spots that we can call SNPs at or markers, informative markers through whole genome sequencing. And this is just a little project that I had. Some of these sheep, so these are your sheep, right? So some of them came from Virginia. And this is why I'm saying that your cluster will be small. Because
right.
when I look at these other breeds of sheep, now this is a composite breed, this is a composite breed, hemps,
right.
Not really, right? I can tell that the lowest number, that means that you don't have a lot of heterozygosity in this breed. So this data is representative of 10 unrelated individuals that we whole genome sequenced.
Okay.
Add more data, would it change a little bit? I'm sure it would, but I think the overall trend is not going to change. Okay, so these are all US sheep.
Okay, thank you.
So these, again, I actually looking at different variant collars here. Again, this was a grad student's project. And the other thing I just wanted to show you here is, so this, right, remember dots and eigenvalues that are very not self-explanatory, and those clusters, this is actually flock 54 data. This is not 50K data, right?
Okay.
And the only reason why I'm asking, does flock 54 help you in this regards, is because it's a cheaper product, right? It's about half the price. So do you need to pay twice as much for the 50K? Maybe you do, but I don't think that you do for what you want to do. right? And so this is flock 54. This is basically one breed of sheep here. This is another. This is actually different producers. And what happens is you'll tend to see these are half sibs. They sit right on top of each other. So this is the guy's whole flock. It groups very differently than this guy's flock. And then this guy brought in some different genetics. He actually bought some of these sheep from here. That's why you see the blue dots in there. And then he brought in some different lines. And so this other flock down here is also from this person, but it's a different line. And so depending on how far down you drill into this information, you can actually, I can zoom in and zoom in and zoom in, and I can tell, and then I can tell who all the sires and all of their offspring will all cluster across each other, kind of like this little group right here, right? So if I zoom in and zoom in, this is like one sire's offspring, and then you'll have some other ones. So this is flock 54 data. So you do get some very sort of similar information using that kind of data.
But those were all one breed. That data was all one breed.
So the paternal one that was his flock. That was the entire flock, and you can see actually the two different. One was more of a I think a Ramble. No, it's not. The other one was Rambouillet. The blue dots were Rambouillet, but he had more of a composite breed, and so with his breed, that's how come you see it spread out like that. And he brought some more sheep in, so you can actually, that's how we can actually define who the sires are or who the parents are by how well they cluster. And so I just wanted to give you a little bit of a sense of what that flock 54 data looks very similar. It is not as powerful as the 50K for sure, because it's 1,000 markers versus 50,000 markers. And the 50K chip that we use for the NSIP is actually 40,000. So you're basically 1,000 versus 40,000. So you get better resolution, but it's not for sure. But you pay for that resolution, and how much more do you absolutely need for your question? So that's the right. And so given that I helped develop the Flock 54 product, I was actually going to use money from that grant to pay for all of this. So that would be my thing, would be I get to look and see how well this Flock 54 compares to 50K. How well does it do for this thing, right? So really it costs you got to go out and get the samples, right? But moving forward, remember, I'm a scientist at a university. Right. I'm just trying to help sheep folks understand genetic information, trying to come up with projects for students to do, and things like that. So I can be helpful if I'm not a service provider. I don't sell a service. I don't whatever. I could be helpful to help you guys do other things if you wanted to. So it doesn't take us very long to generate those plots. It doesn't take us any time at all to determine parentage, to do those kind of things, right? Can we do that for you? Yeah, we can. But again, we're not really a service provider. That's not really what we would want to do. My idea would be that we get you guys in a scenario where you can be submitting information and data, and you guys can manage it yourself, right? I'm always happy to help and things like that, and point people in the right direction and do things, right? So that's the one thing that the NSIP does, is that's what it does, right? It's a breeding value more of a thing, then but that's kind of what you're paying for there, right?
So in our case, where we're looking at establishing that plot and kind of having an idea of what our breed looks like genetically, and then finding other sheep and having them tested, would you be able to help us compare the results from that unknown sheep with the flock? Because I don't feel like I would want to be the person doing that and saying, you know, you're a St. Croix, but you're not, based on my background as my totally non-genetic background. You know, so is that something you'd be able to help us with? You know, that to look at the unknown sheep and say yes or no. You know, I'm 95% sure that this is a St. Croix, or no, I'm 95% sure it's not. So.
Yeah, absolutely we can. So again, like I said, you know, I developed this flock 54. It was just a thing that I did for the industry, right? It's not a
Sure.
yeah. And so we can do something, and it's, again, you know, that what I was talking about with the flock 54 versus the 50K, that would just allow you to capitalize on those dollars, right? But aside from that project, if you wanted to do, say I want to, I don't know, somehow get some dollars together, and maybe we wanted to do a number of different sheep, you know, I'm just like I helped with the flock 54, happy to help get things kind of going and get a system so that it's a kind of plug and play thing for you guys to do. And then you manage your people, you do your thing, you do your association, you figure out, you know, I can just put the data in, I can generate the plots, I can show you what those plots mean, I can give you my interpretation of those plots, and then that's really kind of up to you guys. So you know, I can be helpful that way.
Okay.
I don't know if that helps you at all, or what your thoughts are.
I think it does. I mean, I think, Eddie, and you can tell me if I'm wrong, we have a flock we believe is purebred St. Croix. It's fairly large, but it lost its registration. And our first use of this data might be to establish the breed and then test that unknown flock and see if we can bring them back in as registered sheep. I mean, to me, that's the first project we had in mind. Is that right, Eddie? That agrees with you? I don't want to be out there doing this on my own here.
Yeah, that's the issue of Dr. Taft, and
right.
I don't know if we'll bring them back into the original registry or as genetically tested pure sheep in a future registry with some crossbreeds. That again is not Dr. Murdoch's concern, but something we've talked about. But I'd like to, if we do this testing, do we need, the reason I told you some of the history of the importation, do we need to also test sheep in the University of the Virgin Islands flock and Stephen Wilder's flock in Virginia State University and Scott Peterson's in West Virginia to make, and Utah State University, to make sure we're including all the genetics?
I think it's helpful to just to establish how, you know, how broad that breed is, but you don't have to do it kind of all at once. I mean, you can add to that, and you can see how things kind of grow or branch out or seem a little bit, you know. And remember, when you think about that circle, so I was trying to pull up my paper there just really quickly, just to kind of make a bit of a point. I probably only have the word of it and not the pictures. I'm just going to look quickly and try and find it online. So the point I'm trying to make a little bit here is that size of the circle and how tight those dots grouped within a circle might change depending on what you're comparing things to, I guess,
Okay.
is what I'm trying to get at. So remember, I just showed you that other kind of data and said, now if I were to zoom in, I could see the sire and all of his relatives.
Right.
And if I zoom out, I can make those dots sometimes look a little bit closer together. If that, okay, I have the paper here. If I can find pictures.
Is this your paper? Because I have that. I don't
Yeah.
know if I can post it,
Can you
but
share?
I could.
I think you can
I'm not
share.
sure. Well, I think something said you're now the presenter.
I just passed it to you, and you can go
Okay,
to the
so I
bottom
would
and
use
share
that
it.
share content.
Yes.
I think I just did here.
Okay, yep, there we
I
go.
just, and
Well,
I'm
good.
not
By the time I figured it out, we'd be out of time anyway.
good. So you see in this paper, or one of the other images, it shows you so here's these Western Suffolk, and then here's some of the other Suffolk. And then when I compare it, the more comparisons I go to, the smaller those things shrink together, right? So this whole
Right.
thing now becomes a smaller group, right?
Okay,
And part of that
right.
is because these soy sheep are so different, right? So it
Okay.
does you can I guess essentially you can kind of zoom in and zoom out, but some of that is depending on what you're comparing to, right?
Right.
When we take these sheep out of the comparison, then we get something a little bit more like this, right? So if we take these sheep and then these other Asian influenced breed sheep out of the comparison, then we can see here, right? And so here you have your sort of more of the Australian breeds are a little bit different, and then the wool breeds. And so when you can zoom in by just asking about these specific groups. Right, so you can change, I guess.
Yeah.
You do that by adding in samples or removing samples to look at.
I see.
So here we can, yeah.
Huh. Well, it sounds like our association would have to ask ourselves a lot of hard questions about how we would want this to work, I guess, is what I'm gathering.
Yeah, I think it's a good place to start. Is like, what exactly do you want, right? I mean, we can just use parentage to sample all of these animals and say, oh, okay, this is going to be our breed base, and we're starting from here, and you have to be related, right? Or you can do the full 50K, and you can say, okay, this is what our diversity looks like, and this is what we're considering, and you have to be able to fall within a reasonable range of this to be genetically similar. The other thing that I didn't show you in any of this, we can actually do genomic relationship matrix to tell how close animals are genetically to each other. And we can do that. We can do that with both Flock 54 and the 50K, but again, the higher resolution is always a little bit better, but it costs you twice as much money.
Okay. Okay. Those are the questions I have. I feel somewhat better better prepared to make decisions and maybe with the association talk about what we want this to do and what we would you know, how we would select the sheep to test and, you know, those how we might want to interpret the results and actually use them. So I think for the moment, I'm good.
Yeah, I think we had two separate goals. One was, can we identify sheep parentage? And the other one was, if somebody has a sheep that's very questionable in phenotype, can we test it to know whether it's crossbred or not?
Right.
Right.
And I think you can do both of those things with either one of the products. That you can do it probably better with the 50K. It's always better to have more resolution. The issue is, and the reason why we did the Flock 54 is just to make some information cheaper, right? And so,
Right.
and they have brought that 50K product down to about, you know, if we negotiated with Neogen, which we can negotiate with Neogen, what would it cost, and how many animals, and what would that price point look like, and what would the breed association, what are you guys willing to do? So if I were to chip in all my time and labor and analysis for nothing, which I'm happy to do, that's not a big deal, to get more animals sampled, what would that look like for you guys?
And you'd mentioned grants. Is there any money available for this type of study, or would they just have to pick it up where we could?
We try and pick it up where we can. So the Katahdin genetics, that was all paid for through a grant. It's paid through for parasite resistance grant. That thing. So yeah, so you do try and pick it up where you can, right? And as a matter of fact, they got their genotype funded. It was through an organic sheep farming grant. So
Wow.
we look around and we try and find ways to get information into people's hands as reasonably priced as we can. But
I don't know where we look for these sorts of grants. I mean, I'm happy to help write a grant proposal if you know, if I knew where the organic sheep farmers had their listings of, you know, or where grant calls for grant writing occurred. You know, I mean, I don't know, where do we look to find those? Is there a clearinghouse of any kind, or do you just start with the USDA, or where do we look for these?
yeah, there's a couple different mechanisms, but it really depends on what you know, you have to be kind of catering to the questions. And usually grants are set up to answer biological questions, right? So in the case of the Katahdin, it's, can we identify markers that are associated with parasite resistance, right? That was the sample thing on that, right? So then most of those grants are meant to help producers identify solutions to problems. So that in that case, that's how that was crafted.
Okay.
So we had almost had something that was trying to preserve a breed or to enlarge a breed by bringing sheep back in or something.
Yeah, maybe like some kind of genetic heritage perspective or something like that.
I mean, Eddie, the first thing that comes to mind is the Livestock Conservancy, you know, for whatever grant possibilities might be there. But certainly if someone's looking for parasite resistance, you know, the St. Croix, it might be easier to find some genetic marker for that in our sheep, given that it's, you know, a big portion of their viability.
Right.
On that note, I actually have a grant that uses St. Croix for that exact reason, but my grant is not to sample the whole thing. It's actually to do whole genome sampling to help look at those types of traits in that breed.
Okay.
And how
So,
many
and then-
animals?
Go ahead.
How many animals would that be?
So in this particular grant that I wrote, it's actually, I think there's only 3 because it's comparing the St. Croix genome to the Bighorn genome, to the Rambouillet genome, to the, I can't even remember all that, to the Navajo Churro genome. So in that case, it's looking at different genomes and the biological traits that are associated with those different genomes. And of course, St. Croix is very important when it comes to parasite resistance. And so it's definitely in there, actually through Utah State.
Wow.
Yeah, Utah State. So that was Noelle Cockett and her group there. Got some St. Croix and did that. I'm probably not registered. I don't know. I'm guessing.
Huh. Well, if you ever need registered sheep, I'm not too far from you. I will, I can draw blood. I can do a lot of things. So,
Well, maybe we just have to be a little bit more creative in thinking
We do.
about ways to, help kind of dovetail some things, right? So that's essentially what the Katahdin group did, right? So they wrote a grant. We wrote a grant for parasite resistance, and then that data is also useful for other things. So yeah, I'll think about how we can capture, because in this case, the other thing that you need to do is capture your population, sample your population genetics, right?
Or a fair amount of it. I have to say, I know what a lot of Katahdin owners are excited about is not parasite resistance at all. They want those genetic EBVs. That's for weaning weight and fast growth. That's what they're jazzed about. So, okay.
Yeah,
Okay.
and that could, again, it's really this information can be used for multiple purposes, right?
Right.
And it's really, and it's how, so can we answer interesting biological questions in order to seek funding, even though we know that data will also, we know that'll be informative. It's not like you just get to use it for one thing, right?
Right.
That's the great thing about this, you can use it for parentage, you can use it to identify all sorts of things. You can use it for genomically enhanced DVDs if one wanted to do that, but then also breed structure and breed population and admixture, and all those kind of things that we looked at in that paper with that analysis.
Okay. Okay. Well, we have lots to think about.
Yeah, I thank you for your time because that's helped me a lot to understand because that flock 54 was a new term to me and I didn't understand all the background. So that's very helpful along with all the other you told us.
Mm-hmm. And so why did I name it flock? Why do we name it flock 54?
Oh, I didn't know that.
Have you heard of 23andMe?
Yes.
Heard of what?
So are there 54 chromosomes? I didn't even know that. Okay.
There are so.
Okay. I didn't know it. It didn't. I was thinking of the old TV show. Was there something about car 54? That's what stuck in my mind. Okay.
Yeah. So 23andMe, it's how many chromosomes humans have. They actually have 23. They have 2 pairs of 23. So flock 54 actually is the other version. So they have 27 pairs, so 54 chromosomes. So
Okay.
it was named flock 54.
Okay. There's 54 in you, EWE, we can make
Well,
bad jokes all
that was
day.
another one. We should have had you on the naming committee. Absolutely.
Yeah, I'm only good if it's funny. Okay. Wow. Okay. Well, I'm sure we'll have more questions, but I think Eddie and we need to get the directors together and kind of, ask questions about what we want to do with this and how we might proceed next, what our next steps might be.
And let
Right,
them
yeah,
get this presentation.
Yeah, absolutely. Happy to
Yes.
ask questions. Yeah, or answer questions. So yeah, just think about what it is you really want to do. So if it's just hey, I want to know if this animal's can fall within this breed. And that I originally just off the top of my head said, can Flock 54 do that for you? We could set up a test. We could pay for those genotyping to ask that question. Does Flock 54 perform as well as 50K does in this case? And maybe that's a starting point for something. And then you continually add data to that collection set, if you so choose, kind of thing. But that was just a thought that I had because I was projects that I had, and I was trying to, how can I help you guys out a little bit with some money that, yeah.
Yeah. One quick question, I think perhaps when we are looking at sampling animals, the samples don't have to be in the same time frame. In other words, we don't have to sample 1,000 animals today. We could do, we can keep adding to the profile as we do them for, however we get to do them. So, okay.
Yeah, absolutely. You want to, yeah. I mean, there's those databases are, and we can pull information from other things to help add to your database. So I can pull information from my sequence data to add to that so that we know, and I can pull information from other St. Croix studies. There's been a couple other ones, pull information so that you have that in your database as well. And you just continually kind of add to it and track how it, yeah.
Okay, that I wondered if at the University of the Virgin Islands, if they've done anything, if they've sequenced any of their sheep. So, if we choose to add those in, the U.S. flock, I think has been isolated on the U.S. mainland now 50 years. I'm guessing it's about that. Eddie would probably know exactly. So I don't know how much confusion we would add by going back to the Virgin Islands flock and saying, let's look at your sheep as well.
Yeah. Well, I think you can ask that question and sample the genetics and say, okay, how close, how far? Do they group with what we have here? And then are we making progress by doing that? Or is that what we want to do, kind of thing? Or have they went a different way, in a way that you don't want to go anymore or something like that? So,
yeah.
I've got a lot of influence in our flock from a ram that was imported in the 80s.
Okay. USU 1339 is a ram that keeps popping up in a lot of my pedigrees that I see. So I don't know who he was, but he certainly got around. So I don't know where. USU 1339.
There was a USU 1003 that was highly used.
See, even within the US, I think we have maybe distinct populations, maybe. I'm not sure on that, but okay.
Yeah, and that's what I was saying about it depends on how zoomed in or how far out you go, right? So if you zoom in, you can see those little clusters kind of group out together. And then when you zoom out, it might actually still look like a very tight-knit breed in comparison to other breeds and other things, right? But when you zoom in, you'll be able to get right down to the family level.
Right.
Kind of thing. That, yeah.
Okay. Well, I think we have a lot to think about, Eddie.
Yeah, that has been very helpful.
Yes, we appreciate your time. So thank you very much for that.
Yeah, absolutely. Again, let me know if I can any help, and I won't automatically screen emails from stopandgoparty.com. I can't remember what his email is.
Oh, gosh.
DJ Sparky, yeah.
Yeah. Well, let me know if I can, you know, literally be of any help again. You know, I know it costs money to do these kind of things, and you know, and yeah, they're just kind of hard to kind of get off the ground. But I think once you get them off the ground and get them running, they can be, you know, really quite helpful, I think.
Right.
Yeah. Yeah, I think so. Well, I'm
Is that-
pretty sure I'm going to have more questions, so you may be hearing either from me directly my email says at Shasta Ranch, so there's nothing about, nothing like 54 and you, but, or maybe it'll come through Eddie so that you just have one point of contact and don't have, you know, a lot of people asking similar questions or something. We'll work that out on our end there, but I'm sure those questions will come up. I'm absolutely
Right.
sure. So that's all I have, I think,
Okay.
for right now. So again, thank you for your time.
Yeah,
here.
All right.
absolutely. My pleasure. And it was nice to see you in person, Eddie, and it was nice to meet you, Lori.
Yes,
All right,
indeed. It
thank
was
you.
nice to meet you. So I wish more of our members could have come, but I think sometimes the East Coast time makes it difficult, you know, the time difference. So my whole family's from back east, so I know how that works. You know, I just plan on getting up early. So
I have
work it from there.
I have a project. It's a sheep project that there's some folks from Scotland on the project, so they're at the Roslin Institute, and then I have some folks that are from New Zealand. So,
Wow.
so trying to plan a meeting around both of those. So, you know, so New Zealand's a day off.
Right.
Yeah, and so it's always very interesting, and inevitably somebody's a little bit later and somebody's a little bit earlier than they really wanted to be, and then yeah,
Yeah.
but we do our
Well,
best to accommodate folks. So
yeah. It's the price of a technological wonder like Zoom, I guess. So, okay. Eddie,
We
is
thank
that
you.
everything? Yes,
Yeah,
we
that's
do.
fine.
All right.
Yeah. All right. Thank you very much.
thank
You
you.
bet.
Bye.
Bye.
Do that.